Hi, my name is Shelby Howard. Today I'm doing this video for Bio 225, which is for microbiology. The date is 10/3/2018. Today I'm going to be doing a video on biochemical testing for microbial identification, carbohydrate fermentation testing. Okay, so first off, let me go ahead and just light my candle. Okay, now that my candle is lit, I'm going to put that down. So first off, I went ahead and labeled all of my tubes S. cerevisiae and then the S. epidermis. So, and I have them labeled for glucose, frontal, and meniantal. So first off, let me go ahead and flame the cerevisiae frontal. The cerevisiae glucose first. Let me go ahead and do that one. So let me just pass the lip with the flame, and I already let my these sit for approximately half hour. So that they'd be ready for this part of the experiment. And so I'm going to pour half of the glucose into this jar, this tube. Okay, there's that one. Let me close this. And let me go ahead and sterilize that again before putting the cap back on. I'm going to twist the cap back on, shake this one so that the powder dissolves. The powder all looks like it has dissolved. So I'm going to go ahead and take the next one. That's the one S. epidermis, which is for glucose as well. Making sure to sterilize that lip. I'm going to pop this open and pour the rest of the powder inside of this one. Okay. Now that all the powder is out, I set this one to the side. I'm going to reflame this one right here and put the cap back on, and I'm going to shake it again just so that the powder is mixed in there. Okay, now I'm going to do the same thing, starting with the number 2 one, which is going to be for fructose, and I'm starting with the S. cerevisiae. Making sure it's sterile. So you go ahead and grab the fructose. Okay, cool. So let me put the rest of this off to the side. I'm going to sterilize the lip. Sorry about that, my goggles started to fog up a little bit. I'm going to recap this one. And go ahead and shake it for the powder that's in there. I'm going to grab the next tube, which is for the number 2 S-epidemians, and we're going to sterilize the rinse. And then I'm going to pour the rest of the glucose in here. I mean the fructose. Fructose is going into the number 2 ones. Oops, sorry. Well, let me sterilize that again really quick. Grab this one and shake it really well. And I'm going to repeat the same thing, putting this one off to the side. For the last one, which is the monotonial, okay, so now I'm going to go ahead and flame that tube now that I have the powder inside of the broth. I'm going to go ahead and recap this one and shake it to make sure that powder is nice and in there. Now I'm going to go ahead and pour the rest of this powder after I flame my second tube, which is the S-epi, is what I have it labeled as right now. Okay, now that all of that is in the tube, let me go ahead, flame the tube. Going to shake it. Now they all have the correct powder in each, because I just took the amount provided and put half in each. So let me go ahead and go back through and make sure that I shake all of my vials well. Just so that's all nice and mixed in there. Now, as those are all starting to settle, I'm I'm going to go through again and I'm going to sterilize them and put the, they're called the German tubes. Yeah, I'm going to put a Durham tube in each one with the opening down at the bottom. So let me start with this one. Just so that everything's still sterile. Okay, I already went ahead and placed them in alcohol and let them dry. So I'm dropping my first one in. And, let me go ahead and re-sterilize this. So the lab manual says that if it's catching air and it's sticking up a little bit, just to rotate it over and then turn it back. And the air should the air bubble will dissipate. Which now it seems like there's not an air bubble in this one. So let me go ahead and do the same thing with the next one. Let me go ahead and flame the top to sterilize it. I'm going to grab my next one and drop it in there. I'm going to do the same thing because it does seem to be catching an air bubble. And I'm going to turn it over and then turn it back. And then I'm going to do the same thing and just keep adding the Durham tubes inside of my other tubes. And then there's an air bubble, so I'm going to turn it over, let that air get out, and then drop it back in. Starts to settle. I'm flaming the next number 2 one as well. And I'm going to place another one in here. I'm repeating the steps and making sure that the opened end is facing down towards the bottom. Wait, I didn't sterilize that one again. Hold on. Let me sterilize it one more time before placing that cap back on. Doing the same one with my number 3 tubes. Again, having dang it, I don't know why I keep trying to put the lids on without sterilizing them. We're in week 5. I should be used to sterilizing everything by this point, so again, I'm going to try to get the air bubble out. There we go. So the air bubble's out, it's sunk. And finally, we're on our last one. Okay. So now I'm going to grab my last tube. Again, try to put on the cap, but I'm going to sterilize it. Now the air bubbles going. Place that one back in here, and I'm going to check to see if the other ones still have little air bubbles. Okay. And these ones don't appear to have air bubbles inside the German tubes. Okay. So now I'm going to go ahead, and I already cleaned my pipettes, and they've already been sterilized with the alcohol, and they've already been set to dry. So now I'm going to go ahead and take all of my S. cerevisiae tubes, and I'm going to open them, and I'm going to pipette 4 drops of the S. cerevisiae into each tube. Okay, sorry about that delay. I'm going to go ahead and grab my nutrient broth culture of the S. cerevisiae, and I'm going to go ahead and sterilize it. 1 second. Okay, so I'll go ahead and unscrew it and flame it to sterilize it. I'm going to place this one standing right up on the counter. I'm going to go ahead and open all of my other red cerevisiae tubes and set them inside of the tray over here so that they don't fall over. Because I'm going to go ahead and take my pipette and pipette 4 drops into each tube. Each of the cerevisiae tubes. So I already have my pipette, it's sterile. I'm going to go in, making sure not to touch the sides or anything. And then I'm going to drop 4 drops. One, 2, 3, 4. Okay, so I'm going to put the rest back in my cerevisiae tube. Now I'm going to set this pipette off to the side because I already went ahead and did all 3. Because that pipette's going into my bleach bucket. I'm going to re-sterilize my nutrient broth tube with the S. cerevisiae in it so that I can use it for future use. And now I'm going to screw each of the lids back on. My tubes. And then I'm going to let these incubate for 12 hours. But let's first go ahead and finish the other side. So I'm going to now take the tops off of the S. epidermis side. And I'm going to place them back here in the rack so that you can see them. And I'm making sure that I keep my lids separate so I don't put on any of the wrong lids. I'm going to go ahead and take my nutrient broth that's labeled S. epidermis, and I'm going to go ahead and put that one through the flame to sterilize the tube. Okay, now that that's sterile, I have my pipette already ready to go. So now I'm going to place my pipette in, grab some of the broth, and I'm going to place it 4 drops into each 2, 3, 4. 1, 2, 3, 4 into each one, and I'm going to put the rest back in here. I'm going to place this in my bleach bucket. I'm going to go ahead and sterilize my nutrient broth one more time. And I'm going to close it. I'm now going to recap all of the tubes that my epidermis tubes right here, so that we can go ahead and set them in incubation for 12 hours. And if I don't see any changes of color or anything like that, and fermentation wise, I'm going to go ahead and let them sit for another 6 hours. So thank you for watching my video, and I'll see you in 12 to 18 hours.